The immune system consists of factors that provide innate and acquired immunity and has evolved to become more specific, complex, efficient, and regulated. One of the principal functions of the human immune system is to defend against infecting and other foreign agents by distinguishing self from non-self and to marshal other protective responses from leukocytes. The immune system, if dysregulated, can react to self-antigens to cause autoimmune diseases or fail to defend against infections. The immune system is organized into discrete compartments to provide the milieu for the development and maintenance of effective immunity. Those two overlapping compartments, the lymphoid and reticuloendothelial systems, house the principal immunologic cells, the leukocytes. Leukocytes derived from pluripotent stem cells in the bone marrow during postnatal life include neutrophils, eosinophils, basophils, monocytes and macrophages, natural killer cells, and T and B lymphocytes. Hematopoietic and lymphoid precursor cells are derived from pluripotent stem cells. Cells that are specifically committed to each type of leukocyte are consequently produced with the assistance of special stimulating factors. Cells of the immune system intercommunicate by ligand-receptor interactions between cells and/or via secreted molecules called cytokines. Cytokines produced by lymphocytes are termed lymphokines, and those produced by monocytes and macrophages are termed monokines. Cells of the lymphoid system provide highly specific protection against foreign agents and also orchestrate the functions of other parts of the immune system by producing immunoregulatory cytokines. The lymphoid system is divided into 1. Central lymphoid organs, the thymus and bone marrow, and 2. Peripheral lymphoid organs, lymph nodes, the spleen, and mucosal and submucosal tissues of the alimentary and respiratory tracts. The thymus instructs certain lymphocytes to differentiate into thymus-dependent lymphocytes and selects most of them to die in the thymus and others to exit into the circulation. T lymphocytes circulate through the blood, regulate antibody and cellular immunity, and help defend against many types of infections. The other classes of lymphocytes, B cells and natural killer cells, are thymic independent and remain principally in peripheral lymphoid organs. Cells of the residential provide natural immunity against microorganisms by 1. A coupled process of phagocytosis and intracellular killing, 2. Recruiting other inflammatory cells through the production of cytokines, and 3. Presenting peptide antigens to lymphocytes for the production of antigen-specific immunity. The residential consists of 1. Circulating monocytes, 2. Resident macrophages in the liver, spleen, lymph nodes, thymus, submucosal tissues of the respiratory and alimentary tracts, bone marrow, and connective tissues, and 3. Macrophage-like cells including dendritic cells in lymph nodes, Langerhans cells in skin, and glial cells in the central nervous system. Leukocytes, the main cells in the immune system, provide either innate or specific adaptive immunity. These cells are derived from myeloid or lymphoid lineage. Myeloid cells include highly phagocytic, motile neutrophils, monocytes, and macrophages that provide a first line of defense against most pathogens. The other myeloid cells, including eosinophils, basophils, and their tissue counterparts, mast cells, are involved in defense against parasites and in the genesis of allergic reactions. In contrast, lymphocytes regulate the action of other leukocytes and generate specific immune responses that prevent chronic or recurrent infections. These are one of the major types of cells that are recruited to ingest, kill, and digest pathogens. Neutrophils are the most highly adherent, motile, phagocytic leukocytes and are the first cells recruited to acute inflammatory sites. Each of their functions is dependent upon special proteins, such as the adherence molecule CD11b/CD18, or biochemical pathways, such as the respiratory burst associated with cytochrome B558. Eosinophils defend against many types of parasites and participate in common hypersensitivity reactions via cytotoxicity. That cytotoxicity is mediated by large cytoplasmic granules, which contain the eosinophilic basic and cationic proteins. These cells and their tissue counterparts, mast cells, produce cytokines that help defend against parasites and engender allergic inflammation. These cells display high-affinity surface membrane receptors for IgA antibodies and have many large cytoplasmic granules, which contain heparin and histamine. When cell-bound IgA antibodies are cross-linked by antigens, the cells degranulate and produce low molecular weight vasoactive mediators through which they exert their biological effects. Monocytes and macrophages are involved in phagocytosis and intracellular killing of microorganisms. Macrophages process protein antigens and present peptides to T cells. These monocytes/macrophages are highly adherent, motile, and phagocytic. They marshal and regulate other cells of the immune system, such as T lymphocytes, serve as antigen processing presenting cells, and act as cytotoxic cells when armed with specific IgG antibodies. Macrophages are differentiated monocytes, which are one of the principal cells found to reside for long periods in the RS. Macrophages may also be recruited to inflammatory sites and be further activated by exposure to certain cytokines to become more effective in their biologic functions. These cells provide efficient, specific, and long-lasting immunity against microbes and are responsible for acquired immunity. Lymphocytes differentiate into three separate lines: thymic-dependent cells, or T lymphocytes that operate in cellular and humoral immunity, B lymphocytes that differentiate into plasma cells to secrete antibodies, and natural killer cells. T and B lymphocytes are the only lymphoid cells that produce and express specific receptors for antigens. These cells are involved in the regulation of the immune response and in cell-mediated immunity and help B cells to produce antibody. Mature T cells express antigen-specific T cell receptors (TCR) that are clonally segregated. Every mature T cell also expresses the CD3 molecule, which is associated with the TCR. In addition, mature T cells display one of two accessory molecules, CD4 or CD8. The TCR/CD3 complex recognizes antigens associated with the major histocompatibility complex, MHC, molecules on target cells. The TCR is a transmembrane heterodimer composed of two polypeptide chains. Each chain consists of a constant and a variable region and are formed by a gene-sorting mechanism similar to that found in antibody formation. The repertoire is generated by combinatorial joining of variable, joining, and diversity genes and by N-region diversification. Unlike immunoglobulin genes, genes encoding TCR do not undergo somatic mutation. Thus, there is no change in the affinity of the TCR during activation, differentiation, and expansion. These cells are the primary regulators of T cell and B cell mediated responses. They 1. Aid antigen-stimulated subsets of B lymphocytes to proliferate and differentiate toward antibody-producing cells. 2. Express the CD4 molecule. 3. Recognize foreign antigen complexed with MHC class II molecules on B cells, macrophages, or other antigen-presenting cells. And 4. Aid effector T lymphocytes in cell-mediated immunity. Currently, it is believed that there are two functional subsets of T helper Th cells. Th1 cells aid in the regulation of cellular immunity, and Th2 cells aid B cells to produce certain classes of antibodies. The functions of these subsets of Th cells depend upon the specific types of cytokines that are generated, for example interleukin-2, IL-2, and interferon gamma, IFN gamma, by Th1 cells and IL-4 and IL-10 by Th2 cells. These cells are cytotoxic against tumor cells and host cells infected with intracellular pathogens. These cells 1. Usually express CD8, 2. Destroy infected cells in an antigen-specific manner that is dependent upon the expression of MHC class I molecules. These cells suppress the T and B cell responses and express CD8 molecules. NK cells are large granular lymphocytes that nonspecifically kill certain types of tumor cells and virus-infected cells. Killing by NK cells is enhanced by cytokines such as IL-2 and IFN gamma. NK cells are also activated by microorganisms to produce a number of cytokines. These circulating large granular lymphocytes do not express CD3, TCR, or immunoglobulin, but display surface receptors, CD16, for the Fc fragment of IgG antibodies. These cells differentiate into plasma cells to secrete antibodies and are involved in processing proteins and presenting the resultant peptide antigen fragments in the context of MHC molecule to T cells. The genesis of mu and delta chain positive, mature B cells from pre-B cells is antigen independent. Pre-B cells in the bone marrow undergo gene rearrangement for IgM heavy chains and consequently express those proteins in the cytoplasm, but no immunoglobulin light chains. B cell development is characterized by recombinations of immunoglobulin H and L chain genes and expression of specific surface monomeric IgM molecules. At this stage of development, B cells are highly susceptible to the induction of tolerance. Once these cells acquire IgD molecules on their surface, they become mature B cells that are able to differentiate after exposure to antigen into antibody-producing plasma cells. The activation of B cells into antibody-producing/secreting cells is antigen-dependent. Once specific antigen binds to surface Ig molecule, the B cells differentiate into plasma cells that produce and secrete antibodies of the same antigen-binding specificity. If B cells also interact with Th cells, they proliferate and switch the isotype of immunoglobulin that is produced, while retaining the same antigen-binding specificity. This occurs as a result of recombination of the same IgVDJ genes, the variable region of the Ig, with a different constant region gene, such as IgG. In the case of protein antigens, Th2 cells are thought to be required for switching from IgM to IgG, IgA, or IgA isotypes. IgM is therefore the principal antibody produced during a primary immunization. This primary antibody response is manifested by serum IgM antibodies as early as 3 to 5 days after the first exposure to an immunogen, peaks in 10 days, and persists for some weeks. Secondary or anamnestic antibody responses following repeated exposures to the same antigen appear more rapidly, are of longer duration, have higher affinity, and principally are IgG molecules. When antibodies bind to antigens, they may first neutralize pathogenic features of antigens such as their toxins, 2. Facilitate their ingestion by phagocytic cells, 3. Fix to and activate complement molecules to produce opsonins and chemoattractants, or 4. Participate in antibody-dependent cellular cytotoxicity. In addition to antibody formation, B cells also process and present protein antigens. After the antigen is internalized, it is digested into fragments, some of which are complexed with MHC class II molecules and then presented on the cell surface to CD4+ T cells.